Okay, so now that we've discussed abundance and distribution of organisms, we next move on to the other consideration of ecosystems, which is energy. Energy in ecosystems, how it gets in, how it moves through the ecosystem. Okay, so luckily, this is not going to be anything brand new. We've already discussed the idea that all cells, in order to function, they need to have ATP. And we've discussed that ATP is produced when complex organic molecules are converted into simple inorganic molecules. Okay, so that's the process of respiration. Respiration allows cells to convert complex organic molecules such as glucose into simple inorganic molecules such as carbon dioxide and water, and the production of ATP for use by the cell to maintain its metabolic reactions and processes and other things needed for life. However, problem being that where do these complex organic molecules come from? So we've already discussed that complex organic molecules are actually made from simple organic molecules. Okay, simple organic molecules are used to make complex, or simple inorganic molecules are used to make complex organic molecules. Essentially, carbon dioxide and water are used to make glucose, and then the glucose is used in respiration. Okay, the process by which we can convert these simple inorganic molecules into complex organic molecules is photosynthesis. Okay, so the idea is that organisms called producers, or we might call them autotrophs, are able to make these complex organic molecules using energy in some form from their environment. Okay, and this is essentially what we discussed before, and the idea that the complex organic molecules that they produce, they use for respiration. So the producer itself breaks these complex organic molecules back down to during respiration for its own production of ATP, but any organism that's around can consume the plant or the producer itself directly and get access to these complex organic molecules. So we are talking about a consumer here. So the consumer can also consume the autotroph, get access to its complex organic molecules, and again use these molecules as a kind of fuel for respiration. And in that way, the consumer, be that a primary consumer that eats the plant directly, or even a secondary consumer that eats the first consumer, all of them use their whatever way they can to get to these complex organic molecules to carry out respiration and produce ATP for themselves. And in so doing, they are reconverting the complex organic molecules back to the simple inorganic molecules. Okay, so that's the story. We already discussed this in the first section. Now we really look in detail at what this process of photosynthesis actually is, and later on in the year, when we come to topic 7, we'll look at the process of respiration in detail as well. Okay, so the first slide is basically a review of what we've just discussed. the idea that ecosystems are made of communities of organisms, and the reason we have these communities of organisms, at least one of the reasons, is because it sets up a flow of energy. So the producer produces these complex organic molecules, and essentially the other organisms have evolved to take advantage of these complex organic molecules. So if you're a herbivore, you eat the producer directly, but if you're a consumer, you consume the herbivore that has built its own complex organic molecules from the complex organic molecules that it got from the producer. So in any way you look at it, all these organisms are just essentially finding a way to get at these complex organic molecules so that they can put them through respiration and produce ATP. But what we are concerned with right now is how did those complex organic molecules come to be? So just a quick recap of what autotrophs do. Remember, autotrophs are doing this. Change the color here. Autotrophs are doing this. Okay, they are taking small molecules that they have at their disposal, CO2, H2O, water, okay, and they are putting them together to make larger, more complex molecules with a store of chemical potential energy. But energy doesn't come from nowhere, so it must come from somewhere, and this energy it takes to join these small molecules up into these larger ones. So it takes energy in order to do that, for example, to convert CO2 and water into glucose, as what photosynthesis does. But the reason why there's photo in the photosynthesis is because the photo is the source of the energy it takes in order to convert carbon dioxide and water into glucose. Okay? Whenever you go from small molecules to larger molecules, there must be an input of energy in order to do that, because the number of bonds is going up. There's energy in them bonds. Okay? So if you want to build something with more bonds, i.e. what this is, compared to some things that have less bonds, you're going to need to input energy. And that's why any autotroph uses energy from its environment. Now photoautotrophs use light energy. Photoautotrophs use light energy in order to make the conversion of smaller molecules into larger molecules. But other organisms use other types of energy, and they are not called photoautotrophs. Okay? Anyway, all organisms, including plants, they also need to do the other thing, which is, so even though only autotrophs do this thing right here, all organisms need to do this, which is to break down these larger molecules into smaller molecules again, so that we can have access to the energy. But the energy is not the same. The energy is not the same. The energy released from these processes, that energy is used to produce ATP, smaller packets. Okay, so small packets of energy, ATP, and this is used to fuel cellular processes. Okay, so let's move on. Types of producers then. So we've said all producers use some form of energy to convert simple inorganic molecules into complex organic molecules, and generally speaking, there's two types of producers which use different types of energy to do this. We have the first one, which is the plants, the algae, and some bacteria that can photosynthesize, i.e. they are using light energy from their environment to convert these simple inorganic molecules into complex organic molecules. But in some places on the Earth where there's not a lot of light available, it hasn't stopped ecosystems from developing. And the reason why is because there have been some bacteria that have evolved a way to use chemical potential energy rather than light energy in order to make their complex organic molecules. Now, no kind of implication as to which one evolved first. I have a feeling it would have been the chemosynthetic organisms, but I'm just guessing. Yeah, so chemosynthesis happens in the bottom of at very low kind of depths of the ocean where there's not hardly any light is available, and so these ecosystems have evolved because the producers here don't use light energy to make their complex organic molecules. They are able to make their complex organic molecules, as you can see here, from simple inorganic molecules, as you can see there. However, the energy they use to do that is chemical potential energy. So they use the chemical energy from other chemical reactions in order to drive the production of the complex organic molecules. Okay, so these types of producers are called chemosynthesizers, whereas the types of producers that we see around us on a daily basis, they are called photosynthesizers. Okay? Anyway, in any case, what we are talking about is the same thing. Okay? So you have these producers, whoa. Okay. So you have these producers, and what they are doing is getting energy from the environment into the ecosystem via their complex organic molecules, these ones right here, and once it gets into the producers, that energy in the complex organic molecules then moves through the other organisms in the ecosystem. Okay? So it doesn't really matter where the energy comes from. Okay, so let's look at photosynthesis now. The way in which light energy is used, okay? The way in which light energy is used to convert simple inorganic molecules to complex organic molecules. Let's look at the mechanism. A lot of our year one things that we learned, concepts about proteins, about enzymes, chemical reactions, a lot of that is going to be applied here. So you might want to look over that before you begin. Okay, so this is the overall process. I'm sure you remember from GCSE. Our carbon dioxide, simple inorganic, water, simple inorganic, oxygen, simple inorganic, but glucose, complex and organic. Okay, so that takes us to glucose as a process of photosynthesis is right here. Okay, but the bigger idea here is this, which is, what does the plant do with glucose? Okay, plants are not simply lumps of glucose with a few enzymes. Okay, that wouldn't work. Effectively, what we are looking at here is this bigger idea that if you have one cell, okay, one cell, one plant cell, it's got chloroplasts. Okay, this is very, very important, so please pay attention. One plant cell can photosynthesize. It makes lots of glucose. Excellent. But that glucose is not the end point. Yes, some of that glucose is used in respiration and is broken down back into CO2 and water, and we get ATP. Okay, so that's one part of it. But the other very important part of it is this, that glucose that is produced by photosynthesis is used to make the other important molecules that plants are made up of. Starch, lipids, amino acids, nucleic acids, we could include cellulose in there. Anything that the plant is made up of, ultimately you could probably trace its roots back to the production of glucose. Okay, starch is very straightforward, you join the glucose up. Lipids, not as straightforward, but the carbon atoms that make up the lipids ultimately came from glucose. And the amino acids, they are only there because they were converted from the carbon atoms that were used to make glucose and they were used to make amino acids. This is a very important idea because it's only when the plant cell can do this, it's got enough kind of resources to make its new organelles, to replicate the DNA, to make new membranes, that one cell can become two cells. Okay? And then by that same principle, how the plant can grow. All right? And what we call that is biomass. All the stuff of which the plant is made, ultimately, all that stuff, all those molecules, all that substance is due to the process of photosynthesis and the production of glucose. Yes, some of it was used in respiration, some of it was broken down, but the excess was stored as starch, used to make lipids, used to make amino acids, used to make new nucleic acids, used to make cellulose in the cell wall, effectively used to build the plant. All right? And then when the herbivore comes along and eats the plant, that's teeth, by the way. When the herbivore comes along and eats the plant, it's consuming those molecules. And in its digestive system, those molecules are broken down back to their constituent monomers, which are still organic molecules. And then those monomers are used to make the molecules that the herbivore needs. So the herbivore's own amino acids, the herbivore's nucleic acids, the herbivore's carbohydrates. Yeah? And then the herbivore, because it's got these raw materials to build its own molecules, it can itself give its cells the molecules that it needs to undergo the process of mitosis, and therefore the herbivore can grow. And then the herbivore can grow and survive and reproduce and so on. But the materials for this comes from the consumption of other organisms if you're a consumer. Whereas if you're an autotroph, the raw materials for growing as an autotroph come from the photosynthesis or the chemosynthetic process in those cells. So this is a key idea, and we're going to come back to that later. Okay, so let's now look at photosynthesis. Now remember, photosynthesis is happening in chloroplasts in the cell. So just for a bit of navigation, we've got our, actually, is it down here? Or up here? Yep, it's up here. Okay, so we've got our plant cells here, and effectively in the plant cell we've got these organelles, and it's here really where the photosynthesis is happening. Okay, the chloroplast. Okay, so we've got the chloroplast here, and that's where the photosynthesis is happening, but it's important to remember, and the chloroplast is indicated here as well, that the chloroplast itself houses two parts. So houses two chemical processes, right? One summarized as the light-dependent reactions here, and the other part is the light-independent reactions. Both have to occur in order to overall convert water and CO2 into oxygen and glucose and other molecules. Okay, so that's the overall process. Water goes in, carbon dioxide goes in, but oxygen comes out, and carbohydrates and other molecules come out. I hope you can see from the diagram, let's get rid of that. I hope you can see from the diagram that it takes these two separate reactions to get the overall reaction that we discussed before, or that you will remember from GCSE. Okay, so let's just quickly summarize this. And if I'm going to summarize it here, if you want a more detailed version, I will give you access to that video as well. I'll put the link in the description. So the photosynthesis process happens in two stages. Okay, in the first one, water is split into hydrogen and oxygen, right? So if we see here, we've got water, and here we've got oxygen. So clearly the hydrogen is what we need to account for, and the hydrogen is now here, guys. Okay, so the hydrogen from this process is now here, and the hydrogen then is needed to keep these light independent reactions going. Okay, so the hydrogen is one thing that we need to extract from water in order to keep these light independent reactions going, these ones right here. Okay, now that on its own is not going to be enough. In the process of extracting water, splitting water and producing oxygen, we also need ATP. So that process going from water to oxygen releases a little bit of energy, and that energy is used as well as the hydrogen to keep these light independent reactions going. By keeping those light independent reactions going, I hope you can see that we are able to convert carbon dioxide, use the carbon in the carbon dioxide, and produce glucose. And in that process, we can also make carbohydrates and other molecules such as lipids, nucleic acids, and so on. Okay? But I hope you can see that we need two things in order to convert carbon dioxide. In order to convert carbon dioxide into glucose and other molecules, we need to provide these reactions with some fuel. That fuel is hydrogen and ATP. Okay, now those ATP and hydrogens we're only going to get access to them, we only produce them if we are able to split water in these light-dependent reactions into oxygen. Okay, so I'm going to describe these processes in a little bit, but this overall idea is very important. It's the splitting of water into oxygen and hydrogen that gives us the ATP, that gives us the hydrogen, that allows us to drive these light-independent reactions. And it's the light-independent reactions that allow us to convert the CO2 into the glucose. Okay? Less complex, remember, more complex. And the way we can, and then once we've got this glucose, we can then make other things from it. This is the big idea. Now let's look at the detail of that. Okay, so before we proceed any further, we're just going to look at where everything is happening. All right, so take-home message is this, that these two stages, the first one is called the light-dependent stage. All will become clear about why it's called that. The second stage is called the light-independent stage. Rather straightforward, everything will become clear. Okay, now let's proceed. this is the structure of the chloroplast. I'm just going to highlight some key bits. It's got an enveloped structure. Okay, so it's got an inner membrane and an outer membrane. So it's said to have an envelope. Okay. Inside, it's got these membranous structures. Okay, so if we looked at the structure of these thylakoids, so if I took one of these disc-shaped things right here, okay, so let's just try and recreate that. This disc-shaped thing right here, that's actually a phospholipid bilayer. Okay, so we've got phospholipids all the way there. That's one layer. Okay, and there's another layer of phospholipids. So that's the membrane structure, okay? And then within the membrane, it's got these proteins. Okay, within this membrane, it's got these proteins, and it's these proteins and pigments that are the kind of the machines that run the light-dependent stage. So this is very important, okay, that these thylakoids are membrane, okay, these thylakoids are membrane structures, and the membrane is what has the proteins and pigments needed for the light-dependent stage. And therefore, it's got this structure that maximizes surface area. Because the more membrane you have, the more surface area of membrane you have, the more you have the components, the pigments and the proteins needed to carry out the light-independent stage of photosynthesis, and therefore the more efficient the whole process can be. Okay? Now, the other important part of this is the stroma, this kind of fluidy, spacey stuff here. So this fluid is called the stroma. And it's the stroma that has the enzymes to carry out the light-independent stage. So the stroma light-independent stage, remember that was stage 2, and the thylakoid, the membrane, has these proteins for the light-dependent stage, the first stage. So the first stage happens in membranes of the thylakoids, and the second stage happens in the fluidy space surrounding it. Okay? So we can go back here. Okay, so that is that. That's the key thing. I mean, there's other things that you should know about, like the more detailed kind of names of the structures there. But in order for us to understand photosynthesis, I'm just going to touch on that. Okay, so during your lesson, you'll go through identifying the structures of the chloroplast in images and so on. Relatively straightforward there. So now let's move on to the light-dependent stage. Dependent stage, remember it's happening inside the chloroplast, within the membranous organelles contained within the stroma. Within the membranous organelles, not organelles, but the structures called thylakoids. Okay, and remember we said that the thylakoids were membranous structures. So this is that membrane that we're talking about, and within the membrane you've got all these components. Okay, so we've got the PS2, PS2 right there. We have got the electron transport chain, then we have got PS1, okay, and then we've got a little bit of another electron transport chain at the end. All right, and this other very important structure called ATP synthase, which we'll discuss what that does. Okay, so generally speaking, overall, this is the light-dependent stage, and remember that what we are looking for is a production of two things. We are looking for hydrogen to be extracted from water, and we are looking for ATP to be produced, because remember, it's these two things that are the products of the light-dependent stage that are needed to keep the light-independent stage going. And remember, the light-independent stage is converting CO2 into glucose and our other complex organic molecules. So very important that that stage happens. So let's begin. Okay, so we start our story here at PS1, and what happens is that light excites an electron. So PS1 contains chlorophyll, the famous photosynthetic pigment. It is, now that you know, it is contained within this protein complex called PS2, photosystem 2. Okay, now photosystem 2 contains the chlorophyll, and it's the electrons in chlorophyll that are excited when light. So when light arrives, it is, the light energy is absorbed by the pigment, and then it kind of focuses in on these electrons, and it excites the electrons so that they go up the energy level. I'll use a different color. Okay, so the electrons get excited and their energy level increases. Now, the excited electron then leaves photosystem 2. Okay, the excited electron leaves photosystem 2 and goes through the electron transport chain. And I hope you've been following the movement of this electron, because the reason why it's shown to be going down in height is because it is losing energy. And so what happens is that it starts off at a low energy, it absorbs the light energy and then goes to a high energy state. In its high energy state, then it goes to the electron transport chain. And it loses energy at this point. Where did the energy go? The energy went in pumping these protons. These protons right here, which are in the stroma, are being pumped against their concentration gradient into the thylakoid space. So this is why you need to know the structure of the chloroplast, so that you can refer to these locations. Okay, so the protons are pumped from here into the thylakoid space, and it's the energy of the electron that was used to make that happen. Okay, so this is effectively like a energy requiring transport motion, but we don't call it active transport because technically no ATP was used, it was the energy of the electron. Okay, so we call that pumping. So the protons were pumped from the stroma into the thylakoid space, and they then go to PS1. So now we are here. The electron is here. The electron's there, and it's arrived at photosystem 1. Here again, more light is absorbed by the photosystem because it's the photosystem that contain the pigments, you see. All that green stuff, and whenever you look at a tree and it looks green, it's because it's got a pigment called chlorophyll. Where is all that chlorophyll? It's in these photosystems. What is it doing? It's absorbing energy to excite electrons. Why? Because of what's happening right here. So the energy is being absorbed, and the electron, as you can see again, it's at a low energy level, but it's excited back up again. Okay? So now it's there because another batch of light energy has been absorbed, and this electron then continues its movement through another electron transport chain until it's joined with this coenzyme, this molecule right here. Okay, it's called a coenzyme because it's not an enzyme itself. It's simply transporting things to enzymes. Okay, so it's a molecule, it's a carrier molecule, and that coenzyme, that carrier molecule, NADP, is carrying this electron. So this electron combines with a proton from this space to give us NADPH. So effectively, this NADP has a hydrogen. Okay, so this hydrogen, right, is being carried by NADP. So I hope you can see how that process allowed us, or that as that process happens, effectively, the more and more that process happens, the more and more H plus protons we get into this thylakoid space. That builds up a concentration gradient where we've got a high concentration of protons in here. Okay? And because the protons are being pumped from the stroma into the thylakoid space, the concentration of protons out here is really small in the stroma. And so the protons, there's a force that is acting on these protons because of that concentration gradient, right? So the protons have got this stored potential energy because there's a tendency for these protons to move down their concentration gradient. Okay, so and when they do that, they release a little bit of energy every time they do that, because effectively they're restoring balance to the universe, right? Because this is a very unbalanced situation, so there's a store of energy there which is going to be released when everything goes back to normal. So, which is effectively what all kind of diffusion is. So, as these protons are at a high concentration gradient, as long as you provide a route for them to go out, they will have a tendency to do that, and that's what this ATP synthase does. This ATP synthase is an enzyme that does a very important thing. It is able to convert ADP into what we want, ATP. Right? Remember, ADP is adenosine with 2 phosphate groups. ATP is adenosine with 3 phosphate groups. So how do we go from this molecule with 2 phosphate groups to a molecule with 3 phosphate groups? We add a phosphate group. Okay? So this is what this ATP synthase does. It takes ADP, takes the phosphates, puts them both together because it's an enzyme, and it produces ATP. Great. Problem. It can only do this when H plus ions are moving through it. Okay? Now this is not a pump. It's not using any energy to do this. Okay? Or in fact so it is using some kind of energy to do it. But the energy is coming from the movement of these protons. Okay? So as these protons are at a high concentration here, and because they're at a low concentration in the stroma, they are moving through ATP synthase from a region of their high concentration to a region of their low concentration, and in the process driving ATP synthase to convert ADP into ATP. All right? So this process is called chemiosmosis, the synthesis of ATP using the proton gradient. Okay? Now, that's a lot of stuff to take in. So, and we're still not done. So, we'll just recap that. Light excited the electron. The electron moved through the electron transport chain. By moving through the electron transport chain, it allowed protons to be pumped into the thylakoid space. This built up a high concentration of protons on the inside of this structure in the thylakoid space, which sets up a concentration gradient. And this concentration gradient, as the protons then move down that gradient through the ATP synthase, it allows ATP synthase to convert ADP plus phosphate into ATP. Okay, so you see the sequence of events all required, okay, all needed in order to make ATP. So, and we can see the dependence on light. If there's no light, the electron in the first place, it doesn't get excited. If the electron doesn't get excited, it does not move through the electron transport chain. If the electron transport chain does not have electrons moving through it, there's no pumping of protons. If there's no pumping of protons, we don't have an electrochemical gradient. If there's no gradient, there's no movement of H plus through ATP synthase. If there's no movement of protons through ATP synthase, there is no production of ATP from ADP and phosphate. Okay, so that is that. Almost. Because what we need to explain now is if these electrons are to keep moving through, obviously chlorophyll doesn't have an endless supply of electrons. For every electron that is lost to the electron transport chain and then ends up here, for every electron that does that, an electron needs to come from somewhere. Okay? And this is where we wrap our story up. Okay? Just like any good movie, we wrap our story up by explaining where the replacement electron comes from. So this is where we turn our attention to water, because right at the beginning we said we needed to split water to produce oxygen. Okay? And this, this was one of the things that allowed us to have the photosynthetic process as part of our equation. And this is the significance of that. Like, this is why you need to split water in order to make glucose. Because remember, glucose doesn't happen without stage 2. Stage 2 doesn't happen without ATP and hydrogen. And those things aren't produced without electrons. And so where does the constant source of electrons come from? It's right here. Because if you split water, remember water is 2 hydrogens and oxygen, right? When water is split, you make 1 oxygen and you got 2 hydrogens, okay? But each of those hydrogens can be split into an electron and a proton, okay? And that's really what happens. It's the electron then from that split, it's the electron then that resupplies the chlorophyll with an electron that it lost, okay? And so the continuous excitation of electrons in the photosystem is maintained with the concurrent kind of splitting of water. So for every electron that chlorophyll loses because it's been excited and it goes off into the electron transport chain, for every electron chlorophyll loses, another is supplied by the hydrolysis of water. Okay, so shall we tidy this up and start again? Okay, so let's begin that again. So stage 1, or we can even begin our story right at the beginning of the hydrolysis of water. Okay, so light excites an electron in chlorophyll, and that electron then goes into the electron transport chain. The movement of the electron, so that's stage 1, the movement of the electron through the electron transport chain allows protons to be pumped into the thylakoid space, building up our proton gradient. The electron is then moved on to NADP, forming NADPH. Okay, I mean, that kind of doesn't make sense, but you've got to appreciate that there's protons here. So the proton, which can be united with an electron, gives us the hydrogen. Okay, so that's what happens there. Right, so we're building up our proton, so the light arrives, electron moves through the electron transport chain, the electron that came from chlorophyll. That movement through the electron transport chain allows protons to be pumped into the thylakoid space, building up our electrochemical gradient. The electron then combines with NADP and hydrogen to form NADPH, and this is what will go to the light-independent stage. So that's part one complete. The building up of the proton gradient causes protons to move out through ATP synthase back into the stroma, allowing ATP synthase to convert ADP and phosphate into ATP. Meanwhile, the water is split into oxygen, protons and electrons, while the protons remain in the thylakoid space, adding to the proton gradient, I suppose. The electrons are used to replenish the lost electrons from chlorophyll, allowing the cycle to repeat. That is the that's the light dependent stage. Okay, so remember, we've got our NADPH, which is our hydrogen. It's going to be going off to drive the light independent stage. We've also got ATP, which is also going to go off to drive our light independent stage. And that is the light dependent stage, guys. So we'll move on. Okay, we'll have a bit of a summary here, but we'll proceed. A bit of background information about how the light independent stage was figured out. That will be interesting. But then we move on to really the key part of this, which is what is happening in the light independent stage, otherwise called the Clavin-Benson cycle. Okay? This stage, remember, is happening in the stroma, and we're looking out, it's happening in the stroma, we're looking out for how CO2 will be converted into glucose, and we kind of have to assume that glucose can be converted into other things. So of course it can be used in respiration, but it can also be used to produce other carbohydrates. It can be used to produce lipids. It can be used to produce nucleic acids. It can also be used to produce protein. All right? So we're looking out for how CO2 is converted into glucose. We're also looking out for how ATP and NADPH produced in the light-dependent stage, how they are crucial in this process. So these are the things that we're going to look out for in order for this process to make sense. Okay, so let's begin, and it all begins here with CO2 and this molecule called ribulose bisphosphate, or RUBP. RUBP contains 5 carbons, as shown in this diagram. Obviously, there's more to its structure than that, but it's the number of carbons that's important to us at the moment. It's got 5 carbons in its structure. CO2 obviously has 1 carbon, okay? And they are brought together in a reaction by the enzyme Rubisco. So this is an enzyme, and it's kind of important to understand that what's happening, and what I'll do is just maybe use my highlighter here, is molecules are taking part in reactions, and there are enzymes which are helping those reactions to happen. And it's very important that we understand the difference. So what's happening here is this thing right here, ribulose bisphosphate, is reacting with carbon dioxide, and they're making this intermediate compound. But I hope you can see that this intermediate compound has got 6 carbons, combining the 5 from ribulose bisphosphate and the 1 from CO2, making the 5-carbon intermediate compound. Okay? Now this intermediate compound is then split, okay, broken down into two 3 carbon. It's just snapped in half, effectively. Okay, snapped in half, 2 molecules of this molecule that's called GP right here. Now this two molecules of that is then converted into 2 molecules of, again, 2 of those, 3 carbons. So what we have to understand is that there's some other change in the architecture of this molecule that doesn't necessarily involve the carbon atoms. Okay, so maybe there's a methyl group added or an OH group removed or whatever it is, I'm not really sure. Okay, but this GP molecule is converted into the triose phosphate, and the triose phosphate is then used, some of it is used to make glucose, okay, but some of it is used to regenerate ribulose bisphosphate. Okay, so let's just do that again. Okay, so we've got carbon dioxide, 1 carbon. It comes in and it's joined by the enzyme. Okay, by the enzyme Rubisco. Rubisco is not part of this process. It's just the enzyme that makes one of the reactions happen. Similarly, there's probably going to be an enzyme right here making this reaction happen. And there's an enzyme right here making this reaction happen. But they're not crucial to the understanding of this process. Okay, it's just that Rubisco happens to be the first enzyme that incorporates the carbon dioxide, and therefore it's very important. Okay. Anyway, we've got carbon dioxide and it reacts with this molecule right here, and that reaction is catalyzed by this enzyme Rubisco, and the product of that reaction is this 6-carbon molecule, which is just called intermediate compound. Fine, don't need to know it. So this intermediate compound is then further changed to produce glycerate 3-phosphate. It's effectively that 6-carbon molecule is broken down to 2 molecules of glycerate 3-phosphate. That glycerate 3-phosphate then is converted into 2 molecules of triose phosphate. Some triose phosphate is used to make glucose and other molecules, whereas some of the triose phosphate is also used to regenerate ribulose bisphosphate, and so the process can continue with more carbon dioxide. And so we can continue making triose phosphate, and so we can continue making glucose and other molecules. So that's basically, the following the carbon atoms. Now, this first reaction here, this is important because this is called carbon fixation. Okay, it's where inorganic carbon is converted into organic carbon. It's very important, and that's why this enzyme is given special prominence. Because if this enzyme doesn't work properly, then the whole thing falls apart. Okay, well, the same could be said for all other things, but it's, okay then, so we've got carbon fixation, and so we've got inorganic carbon converted to organic carbon, very important. But then, remember we said we were looking for carbon dioxide to glucose. Well, I think we've looked at that. We've looked at how carbon dioxide is converted into glucose. Now, the next thing we need to do is to see, well, how is ATP and NADPH involved in that? Right? We said that this process could not happen. We've said that this process could not happen without the input of the ATP and NADPH from the light-dependent stage to stage 1. So how is it that this process needs those things? Well, this is where I will show you. Okay, so let's look here, reduced NADP, right here. Okay, so reduced NADP is converted back into NADP, which means that effectively NADPH is being converted into NADP. So we've contributed, this hydrogen has been incorporated into the process, into the molecule. So this hydrogen has been added in to this process. So the hydrogen that this NADP collected in the light-dependent stage has now been successfully delivered into this process. And we can see now that glycerate 3-phosphate could not be converted into triose phosphate without reduced NADP. Okay? Because without reduced NADP, we wouldn't have this hydrogen in, and for all we know, that this reaction would not be possible without the input of this hydrogen. Okay? So that's the first thing. Next, there's two incidences where we can see here ATP is being reconverted back into ADP, i.e. energy being used now. So in order to convert GP into TP, we needed ATP, the energy of ATP. So this is where some of the ATP that was produced in the light-dependent stage gets used to convert GP into TP. Okay, so ATP is used there as well. ATP is also used here to convert triose phosphate back into ribulose bisphosphate to complete our cycle and regenerate the molecule that we need to if we want to convert more carbon dioxide into glucose. Okay, so that's the Clavin-Benson cycle. This is the light-independent stage. As we can see, light directly is not involved in any of these steps, so it's called the light-independent stage. Okay, but one could argue it could not occur without the light-dependent stage because, well, we need the reduced NADP and we need the ATP in order to drive some of these steps. What we can do is just go back a few slides and just review the whole process of photosynthesis, just so that overall it makes sense. Okay, so what we looked at was that in the first stage of photosynthesis, in the light-dependent reactions, the light-dependent stage, we need water as our input, and water is hydrolyzed, or not hydrolyzed, but during the hydrolysis of water, water is split into oxygen and protons and electrons. And these protons and electrons eventually combine to give us, remember, NADPH. So the hydrogen from that is carried to the light-dependent reactions via this molecule NADPH. So should we just zoom in on that bit? Okay. Okay. The other thing that's produced in the light-dependent reactions was, remember that the movement of the electrons, so the light hitting the chlorophyll raised the energy levels of electrons, which caused them to move through the electron transport chain. As they moved through the electron transport chain, protons were pumped into the thylakoid space, and as a result of that, we built up this electrochemical gradient of protons, which had a high concentration inside the thylakoid space and a low concentration in the stroma. Because of this gradient, the protons move out into the stroma via ATP synthase, causing ATP synthase to produce ATP. So the other product that we have is ATP. And these go into the light-independent reactions, causing the light-independent reactions to occur. And the main thing that the light-independent reactions do is use CO2 as an input, combine it with some pre-existing molecule, okay, and the enzyme that does that is Rubisco. So the CO2 is combined, and as an output of that process, we get carbohydrates. And from the carbohydrates, we can modify them into lipids, amino acids, nucleic acids, and so on. So all the building blocks of all the mass in the plant comes from that. Okay? However, in doing so, because we're using up the ATP, we reconvert that back into ADP, which goes back to the light-dependent reactions to be converted into ATP by ATP synthase again, and the NADP returns after donating its hydrogen, it returns back to the light-dependent reactions to collect more hydrogen. And so we have that. Okay, so that's a quick review. Remember that all the things you need to understand about this start from the structure of the chloroplast, understanding the light-dependent reactions, what are the main stages of that process, and what are the products of that process that are needed for the light-dependent, independent reactions, and what is the main job of the light-independent reactions, converting CO2 to carbohydrates, and how the light-independent reactions require things, substances produced by the light-dependent reactions. Okay, so that's photosynthesis. Okay, and what we'll do is we'll just quickly review this important idea about what happens to the products of photosynthesis, because there's a bigger idea here that's really, really important, which is that photosynthesis allows CO2 to be converted into carbohydrate. Okay, kind of more immediately, it's glucose. But then, some of the intermediates from the Clavin cycle can be used to produce amino acid, lipids, nucleic acid, DNA and RNA, that kind of thing. So really what we're talking about is that, and all these together is what the cells use. So each cell, and we've already discussed this, the products that come out of photosynthesis, some of that is used for the plant's own respiration so that it can get enough ATP to keep all the processes that it needs and all the cells alive. But all these things, the lipids, nucleic acid, amino acids, the carbohydrates, they can also be used to allow the plant to carry out, go from one cell to two cells to four cells, building new cells, and therefore increasing the biomass of the plant. Because as the plant grows, as it goes from 1 to 2 to 4 to 8 to 16 to 32 cells and effectively grows, as it grows, and when they eventually get to trillions of cells, we can actually view that as a growth of the plant. And therefore, the rate at which the plant is able to do this, we can actually measure as the rate at which the biomass increases. Okay? So the rate at which the plant is able to convert light energy into biomass, because effectively that's what it's doing during photosynthesis, the rate at which it's able to do this, that is what we call its productivity. Okay? Obviously, you have to take away some of the energy that it produces, or some of the glucose that it makes is used in respiration, so that doesn't contribute to the biomass because that gets broken down to CO2 again. But all the excess energy that it doesn't use, so all the excess chemical energy that it doesn't use, it converts into biomass, uses it to grow the plant. Okay? And that is what we call productivity. Okay? And that's essentially the concept that I discuss in this slide, is that if we had a sun here, the sun's energy is going into the plant. And as the plant grows, that energy is being transferred to the molecules, i.e. the biomass of the plant. And then when the zebra eats the plant, that chemical energy is transferred to the zebra when it consumes the plant. So all those molecules then transfer to the zebra, and then obviously the zebra is going to lose some of that energy as for various reasons. So it's going to use up some of that chemical energy in respiration. So, it's going to lose that energy as heat. Some energy might not be able to digest, so it will come out in waste. Okay? But this is what we need to understand, is that the process of photosynthesis is what allows this energy to be A, accumulated in the plant, and then B, transferred to other organisms. Yeah, because remember, there's going to be if I can get this right, there's going to be a lion, going for this zebra, and then when that happens, the energy from the zebra is going to transfer to the lion. Okay, so that's what we're talking about. But that energy originally came from the plant converting the light energy into biomass. That's how all the other organisms in the ecosystem were able to access that light energy, or the energy that was in the light. Okay, that the units of productivity are measured in kilojoules, so that's the measure of the energy per meter squared of, because you need to obviously, 2 meters squared of plant is going to make more than 1 meter squared of plant. So it's measured per meter squared, so per unit area of the plant, as well as time, because obviously the amount of energy accumulated in a year is going to be more than in 6 months. So we also measure the energy accumulated in time, and that gives us an idea of how the rate. So all this is really about rate of energy conversion from light to mass. Yeah, biomass, chemical potential energy. Okay, so that's kind of how the energy flows from the kind of producer to the consumer, or the let's say the herbivore, the primary consumer, and then we have the secondary consumer. Okay, but then what happens when the energy gets to the top organism in the food chain? And that is when the whole idea of the recycling comes about. Or not recycling as much as a kind of continuation of the story. So if we look at this picture right here, the energy is going from the sun to the producer to the first consumer, grasshopper, then the frog, and then the snake, and then the final consumer here. Now, this is not the end, because all those molecules in the hawk, even though the hawk doesn't have a predator or anything that will consume it, there are organisms that will nonetheless make use of the energy in the molecules of the hawk. Eventually, when the hawk dies and its body is there will be organisms that are maybe microscopic, or in the case of the fungi, there might be macroscopic, you might be able to see them. However, there are organisms that will be able to digest, break down those molecules that make up the hawk, break them down, absorb them, and use those molecules for their own respiration. Okay, so it's a continuation of the food chain, just not in the same way as all the other stages occurred, but on a kind of molecular level, it's exactly the same. Okay, so these fungi, even though they don't have teeth and they're not actually hunting down the hawk, when that hawk dies, these organisms are there. They are going to secrete enzymes. The enzymes will break down those molecules. The cells of these microscopic organisms will absorb those nutrients and use those in the same way that the hawk, the snake, the frog, the grasshopper did, i.e., funnel those resources into cellular respiration and get the energy back. Obviously, the carbon dioxide returns back to the atmosphere and we begin again. Okay, right? So not so much recycling of energy, but the recycling of matter does happen, yeah? Okay, so then we look at the efficiency. So we discussed at the, we discussed this idea of productivity. So plants could have, we could measure the rate at which plants, per a unit area, per unit time, how much energy of light are they converting into mass, i.e. to grow. Okay, so there are a number of factors that can affect that. So obviously, the thing that's linking all those factors up is this idea that those factors must be affecting the process of photosynthesis. So for example, light, right? Why is not all of the light that falls on the leaf converted used to make glucose or carbohydrate? So why is not all that energy used for the growth of the biomass? So why is it not 100% efficient? And we just look at this idea here that even at the first stage, you might not get all of the light kind of being absorbed by the chlorophyll. As we can see from this diagram, only 40% of the light that hits the leaf is absorbed by chlorophyll. Some might not hit a chloroplast on its way through and therefore makes its way out. So it went through without hitting any chloroplast. Some is obviously reflected. Why? Because when we look at a tree, we can actually see it, which means the light must have bounced off it and gone into our eyes, right? So some light is being reflected there. And some energies, instead of being absorbed by the chlorophyll in the thylakoid membrane, instead of being absorbed there to excite electrons, before it gets to do that, that energy is absorbed by water, which causes it to evaporate, okay? Which is itself is a very important process, as we saw in topic 4, that this evaporating process is very important for the transpiration. And that in itself is very important to keep photosynthesis going. So it's not a wasted process. This energy that's used to evaporate water causes that water to leave the leaf, allowing the transpiration stream to occur, which maintains the supply of water. Okay, which maintains the supply of water, keeps water moving through the plant. And so, remember, water is one of those key ingredients that we need as inputs for the process of photosynthesis. Okay, because without the hydrolysis of water, we don't get that whole light-dependent stage started. Okay, another reason is here, right there. So we have the frequencies of light which are absorbed by chlorophyll. Chlorophyll being a pigment, it absorbs specific wavelengths of light, not all of them. And as we know, that light, visible light, is made up of a range of different wavelengths which correspond to different colors. And pigments absorb specific colors, and what we see in this graph right here is that chlorophyll absorbs some wavelengths more than others, i.e. not all the wavelengths of the light are used for photosynthesis, okay? So that's another reason why the total amount of energy which is in the light is not used, okay? So, and then we discuss other factors that could affect the photosynthesis efficiency of that process of using light energy to make biomass and growing. What could these other factors be? Well, limiting factors is a very important idea, that if you have a process that needs CO2, but the levels of CO2 are insufficient, then even if you have light, and even if you have enough water, and even if the temperature is just right for all the enzymes, if you don't have enough CO2, it's going to limit how much photosynthesis can happen, what the rate of that photosynthetic process can be, and ultimately then what the overall productivity is going to be. Okay, so there's a number of different factors that could be affecting this process, all the way from the minerals in the soil, the water levels, temperature. All those factors need to be high, or need to be just right in order to have the highest productivity. If even one of those is less or not optimal, then it acts as the limiting factor. Okay? So, and I mean, it's important to then, recognize this when it comes up in any kind of question, and use the idea of limiting, not just use the idea, but actually explicitly say that something is acting as a limiting factor. Okay, and we've got some example questions there. Okay, and then we come back to this idea of gross and net productivity. We've hinted at it, though I didn't use the terms at the time, but we said that CO2, so our inputs such as CO2 and energy are used to make carbohydrates, and carbohydrates is used to make biomass. Okay, right? And this is the photosynthesis process. So carbon dioxide, so photosynthesis uses energy to convert carbon dioxide into carbohydrates, and carbohydrates are used to make the biomass of the plant, which makes the plant grow. Okay, great. Okay, and that biomass, the energy that's in that biomass then kind of effectively fuels the rest of the ecosystem. Okay, however, remember that the plant, no organism does anything to help any other organism. No organism appreciates its role in the ecosystem. Any organism is always trying to exploit its environment for its own needs. And if any other organism manages to exploit that for itself, then that's how the whole ecosystem works, okay? So the plants don't have an underlying appreciation of their role as producers of an ecosystem. The main reason why they're doing all this is to grow and increase their own survival and reproduction chances. However, one of the main reasons why they are doing this also is because of respiration, remember? So the carbohydrates that they are producing, they are producing that so that they can carry out respiration. Everything else they're using to make new cells and to grow. Any excess carbohydrate that they have that is not immediately required for energy purposes is converted into biomass. And so we have this idea that of all the energy that comes in, some is used for respiration, and whatever's left is effectively what goes to add mass to the plant. Okay, so what we call this is gross productivity. So of all the energy that the plant converts, we call that gross primary productivity. That's the total amount of energy that the plant was able to convert into chemical potential energy. But it's going to use some of that in respiration. So we have to subtract the amount of energy that is used in respiration, and what we get at the end we call net primary productivity. So that's the energy that's left over after some has been used for respiration. Okay? So that's basically that idea. And it's important, let me just saw that there, that it's basically the NPP that's available to the other organisms in the ecosystem, okay, to the primary consumers, the herbivores. All right. What we're saying is that we're revisiting the idea that energy flows through the organisms of the ecosystem, but it's not as straightforward as all the energy from one organism goes to another organism. Okay, so if we have the plant here, or let's call it the general thing, we have the producer. Of all the energy that the producer has in its molecules, not all of that is going to go to the primary consumer. Not all of the energy is going to go there. Why? So we've got to think of some reasons. So of the producer, producer is going to use some energy for respiration, for its own respiration, and the consumer is not going to eat or consume all of the producer. Okay? So that's a very important idea, that the consumer might, nibble at some grass, but it's not going to consume so all the consumers are not going to completely wipe out all the producers. So not all of the energy that's in the producers goes to all the consumers. Okay? So there's that. And then even like the primary consumer may consume some producer organisms, but then some of that some of that material that it consumes, some of it's going to be digested and absorbed. Great, that's the energy that makes it into the primary consumer. However, much of that is going to come out of the body as undigestible waste. So those are still molecules. Those molecules were made by the producer. Those molecules were not digested by our consumer, and therefore they're going to be coming out of the body as waste. So whatever material made by the producer that's unable to be digested and absorbed by the consumer is effectively going to be decomposed by other organisms. So some organisms will get access to this energy, it's just not the primary consumer. Obviously, of the energy that the primary consumer does absorb, it uses some energy for itself for respiration. Okay? And then we have our secondary consumer, and the same principles apply there. Okay? So not all of the primary consumer organisms will be consumed by the secondary consumer organisms. Of the ones that do get consumed, some energy is used for its own respiration and growth, and otherwise, at the end of its life, if it doesn't get consumed itself, it will be decomposed by the decomposer organisms. Okay? So these ideas are important. The ideas that not all organisms are consumed, so either those organisms will not get consumed and they will die, or they will get consumed, but then some of their energy still remains in the undigestible material in the feces. Okay? So that's the idea that's very important to understand there. Okay? And that's basically that about energy in ecosystems. Right? So it might be good at this point to, like, once you've listened to all of this and it kind of makes sense, maybe have another look at it through the textbooks to see if the overall concept makes sense, and then have a look for the details. Was there anything that didn't make sense to you? If there was, do get in touch, ask me the questions, and I will try to help.